In this lesson, we are talking about invertebrates. And so we're going to start by talking about some of the introductory kind of general characteristics of invertebrates. So first of all, we often overlook invertebrates, but they actually make up 97% of the animal kingdom. So vertebrates, which are things that have a backbone, like humans and other mammals and reptiles, they only make up 3% of the animal kingdom. And the basic characteristics of all invertebrates is that they lack a cranium, so they don't have a skull, and they lack a vertebral column or a spine. Now, if we look at the taxonomic classification of invertebrates, so let's go back here real quick. We'll talk about domain. So invertebrates are in the domain Eukarya. So they are made out of eukaryotic cells. They are in the kingdom Animalia. And then from the kingdom is where we start to get these kind of branching classifications. So we do have some superphylum, which would be, or superphyla, I guess, for plural. So superphyla. And that's the next level of classification when you're getting kind of more specific. Under the superphylum level, you have the phylum level. And under the phylum level, you have the class level. And remember that the domain is going to be the most broad classification. It's going to include the most number of species. And then we're becoming more specific from there. So in this lesson, we are going to be covering the phyla and the superphyla listed here. And I'm not going to overwhelm you right now, but we will break these up into kind of smaller videos and smaller little lessons to make it a little bit easier to keep track of what kind of invertebrate is what. So we're going to start by talking about the simplest invertebrates. And if we look at this phylogenetic tree here, we're going to be looking at the sponges. Now, this phylogenetic tree includes the metazoa, which are all animals, but then we start to branch. So we're going to follow this branch to take a look at the parazoa, and parazoa are animals that do not have tissues. And then there's only one phylum that makes up the parazoa, and that is the sponges. And sponges are phylum Porifera. So phylum Porifera, it's the only phylum that's in this group that's called the Parazoa. And Parazoa just refers to animals without true tissues. And Porifera makes up these very simple organisms called sponges. And if you've ever used like a natural sponge that you get, sometimes you can get them from like a department store or something like that. They can be used to, clean, or back in the day when sponge painting was really trendy, they would often use the natural sponges for that. And what it was is basically the kind of the dead cells and kind of skeleton, not a real skeleton, but kind of the leftover parts of a dead sponge. Sponges are asymmetrical, and they are the only animals that are asymmetrical. They are the simplest invertebrate. They are all aquatic. And as I mentioned, they are parazoa, which means animals without true tissues. So if we go back to a previous lesson, we talked about embryonic development, which we can see in this picture here. And during embryonic development, some animals form this gastrula. And so this process called gastrulation occurs. But in parazoa, in these sponges, gastrulation does not occur. And it also means that we don't end up with those germ layers. And the germ layers, if you recall, are things like the ectoderm, mesoderm, and endoderm. But remember that in sponges, they do not have these germ layers, and that's because they do not gastrulate. And this picture here shows you a couple of different varieties of sponges. So they come in different shapes and sizes and colors. Sponges do have flagellated larvae. That just means that the larvae have flagella. The adults, however, are sessile, meaning that they do not move. They are non-modal. And you can see that this picture here, this would be an adult, kind of a basic adult sponge here. And you'll see that the structure of it is really designed for water movement. So you can see the movement of water by following these arrows here. So these sponges have these canals and chambers and cavities. You can see that they have these kind of pores in their body wall that water can flow through. And this is all because they don't have true tissues, they don't have organs. And so most or all of their gas exchange, feeding, excretion all happens primarily through diffusion with the water surrounding the organism. As we mentioned before, sponges can come in different shapes and sizes, but they all have the same kind of basic body shape to them. They have a spongocoel, which is this cavity kind of in the middle. They have these ostia, which are these pores that water can flow through, as you see here. And then the osculum is the kind of the opening that the water then flows out of. Although the sponges do not have true tissues, they do have some specialized cells and layers that kind of fulfill functions like tissues. So first of all, the penicoderm is the epidermis, which kind of acts like a skin, but it's very different from the skin of humans, for example. But you can see that on this picture here. It's this kind of yellow layer here that makes up the penicoderm. And it is primarily made up of these cells called penocytes. In the middle of the sponge, you have this area known as the mesohyl. So this is the penicoderm, and this is the mesohyl on this picture here. And this is a jelly-like substance between the penicoderm and the spongocoel. So remember that the spongocoel is this kind of space here. So we're kind of looking at the outer wall in this picture here. And so inside this mesohyl, we have collagenous fibers, and collagen is just a type of protein. And it's got a little bit of stretch and flexibility to it. There are also a lot of different types of cells that are found in this area, and you can see those on this picture here, but we're going to talk about them in a little bit more detail on another slide. So now we're going to talk a little bit more about these specialized cell types. So the penicocyte, this is what makes the penicoderm, which is the kind of like the outer covering, which is kind of like an epidermis or a skin. There are cells, there are oocytes, and so sponges can reproduce sexually using eggs and sperm. So they do have oocytes, which are egg cells. And then amoebocytes. These amoebocytes can actually kind of act like a stem cell, meaning it can differentiate into other cell types, including sperm. The porocytes are cells that line the pores or the ostia. So remember, the ostia are these pores here. And so the porocytes are going to line those openings there. The sclerocytes, sclera means kind of like hardening or skeletal, and it secretes these silica spicules that kind of give the sponge a little bit of structure to it. It also provides the sponge with some protection because, as you can see, it's kind of spiky and it might stick out of the penicoderm. And so if a predator comes along and tries to grab the sponge or bite it, it might get kind of stabbed with those little spicules. It's kind of like fiberglass. If you've ever touched like fiberglass insulation, you get these little like pricklies in your skin that are really irritating. The lymphocytes and collinocytes secrete that collagen. Remember, collagen is that kind of flexible protein that helps to give the sponge some shape, but also a little bit of kind of flexibility to it. And then we're going to talk about the choanocyte more on the next slide. We mentioned that sponges are very simple or basic invertebrates. So they do not have a digestive system, a respiratory system, circulatory, reproductive, or nervous systems. They rely a lot on diffusion, and that diffusion is for things like gas exchange, but also excretion, so getting rid of waste products, and also distributing potentially food particles around. Sponges are filter feeders, and that's where these choanocyte cells come into play. So these are cells. And these cells, as you can see, they have these microvilli, which are these here. And the microvilli, they are really small, like microscopic cell extensions. And then they also have a flagellum, which is a protein extension. And all of these extensions help with a couple of different things. So first of all, it helps to create a water current. So the microvilli and the flagella can actually move. And in moving, they actually help to create kind of this almost like a suction of water to pull water into the spongiocele. And you can see the microvilli that's being shown right here. This is also where food is phagocytized, and phagocytism is a type of endocytosis, where essentially you have a cell and you have a food particle, and what happens is that the cell kind of engulfs that food particle like a little Pac-Man. And then once the food particle is inside the cell, it can fuse with other organelles like lysosomes, which have digestive enzymes associated with them. And that helps to break down the food into nutrients. And so the microvilli especially help to increase surface area, which allows more room for phagocytosis. And so there's just more cell membrane available to phagocytize or take in those food particles. Sponges can reproduce a couple of different ways. They can reproduce asexually by budding, and budding just means that some cells or like a little growth kind of forms on the outside of the sponge, and then it might stay there and just become part of the colony, or it might fall off and then continue to grow and form its own colony. They can also reproduce sexually. They are monoecious, which means that they are hermaphroditic. And this just means that one organism has both male and female parts. And so, as we mentioned, these cells, so the sponge has oocytes, and then the amoebocytes can differentiate into sperm. The gametes, so remember gametes are just the eggs and sperm. These are the sex cells. And they are released into the water, and then external fertilization occurs. So fertilization occurs in the water outside of the organism.